Right, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, and very good evening, everyone. Okay, so welcome back to our SID 3019 special topics in applied chemistry. And as I mentioned, today we will be doing a tutorial like session. Okay, so normally when you have a tutorial, we will print out something and then we'll meet up face to face, and then we will give you the piece of paper and then you will do it. And as you are doing it, we will discuss a little bit. Okay, so that's the common way of doing it. So for today, what I'm going to do is pretty much an adaptation of what I've been doing for all our live lectures, whereby we have a quizzes, questions during the quiz itself, during the lecture itself. But today what we will have is instead, I've prepared about 10 questions also. So if we do have time, we will go through all 10. Otherwise, we will just go through until we have time to actually do it. Okay. So for today's lecture, I hope that you guys can see what I'm showing you guys now. Let me check. Can you guys see this? You can see the 3D code for the attendance. Can you guys see this? Anyone? Alisa, can you see this?
Yes.
Thank you, Mastika. Thank you, Ami. Okay. So today's code is first tute. You can just QR this very quickly and then key it in your phone. All right. So I give you 5 seconds to actually do so. Very quick. All right, 5 seconds. Now, for the first questions, before I actually go into the real ones, okay? So do you have any, I think I activated this. Okay, so please go to your Poll EV app. Okay, go to your Poll EV app. Today is the 12th of 4. And what concept? See this one. Okay, and if you want me to explain any concept that was covered, okay, so you can go to your Poll EV app now, and as usual, pollev.com if you are using web browser /hadifuad049. Okay, just key in whatever you want. What concept, not whatever you want, okay, what concept do you want me to recap or explain further? Okay, do this now. I'll give you guys about 1 minute to actually think about something that you will probably want me to explain a little bit more in the next few weeks of our lecture. Okay, if there's nothing after 1 minute, then we will just straight away move to the tutorial questions. Anything? Yes, no, nothing? If you're okay, you can just type okay. That will do as well. At least I know if you guys are actually following what I'm saying and what I've been trying to share with you guys. Okay? Any okay? Any okay? Or we put none. So everybody put none. Anything? Tak ada ya? Okay, I have 7 responses that says none. Okay, so if there's none, then it's fine lah. Okay. All right, so tutorial number 1. So this is based on, of course, everything that we're going through today is based on lecture number 1 to lecture number 5. Okay, so if you did your study, it will be like very easy. If you don't do any revision, then it might give you some hardship. All right, so question number 1. Question number 1. Polyamide is one of the chemical functional groups that is widely observed in the nature. What is the name of the micromolecule formed via a polyamide bonds? A. Carbohydrate. B. Peptide. C. Lipid. D. Nucleic acid. And E. All of the above. Okay, for those who recently just joined in, if you can go to any web browser, or if you have downloaded the PolEV app, you can always go to polev.com/, key in Hadi 049 to actually participate in this tutorial. Okay, otherwise, because there's no lecture at all, so these are just based on practices. I've prepared questions that we will do, and of course the description or discussion about that particular answer and why that specific answer is the correct answers. Okay, I'll give you guys, there's only 10, 11. There are 20 of you here, or 19, so to say. So the rest of you, if you want to join in, then please use this link. Okay, I have 13 now. 14. Anyone can't join? Oh, you cannot enter. Using your browser or mobile? Okay, so maybe your internet is slow. Okay, that is fine. But you know, just jot down somewhere. I will definitely share this slide as well. Okay, once we finish with our tutorial, I will put this whatever we have here, including all the polls, I will put it on Spectrum. At the same time, the tutorial is recorded. I will also post a link on Spectrum. So this is just a revision for you guys if you need to do it in the future. Okay, all right. So the answer is yes, you are correct. Peptide is the correct answer. All right. So just a little bit of revision on biomolecules. Okay, that is a structure of a carbohydrate. Okay, that one is lipid. That one is peptide. And the other one is nucleic acid. Okay, so the questions, the question asks about what is the micromolecule. So the definition of micromolecule are all the 4 options that is available to you: carbohydrate, peptide, lipid, and nucleic acid. Okay, and the question asks which is what is the name of the micromolecule that is formed via a polyamide bond. And if you recall, polyamide, so amide functional group is a carbonyl and an amine, like that. So that is an amide functional group. So when we have, if you look at here, so these are the amide functional groups. Okay, and polyamide means multiple of the amide functional groups. And if you look at carbohydrate, you only have an ether bond, okay, or glycosidic bond. Lipids, you have a lot of these carbon chain, and nucleic acid, you have a phosphorus link. Okay, all right. Number 2. Which of these is not a naturally occurring amino acids? Okay, which of these is not a naturally occurring amino acid without S? Grammatically wrong. Okay, first one is valine. Second one is ornithine. Number 3 is aspartic acid. Number 4 is proline. And number 5 is selenocysteine. Which of these is not a naturally occurring amino acid? Okay, so 1 minute. 1 minute for you to choose. I have 2 responses now. 3. 4. Okay, 30 more seconds. 12, 13, 14, 15, 16. Okay, 17. All right. So just now, Muzaffar and Alisa, can you guys join now?
Can, Doktor.
Yiwen. Boleh. Okay, thank you. Yiwen, I will share the link again at the end of this session. Okay, at the end of this session, I will share it again. All right. So time 10 more seconds. Okay, 5, 4, 3, 2, 1. All right. So this question has 2 answers. Okay. So if you choose ornithine, and if you choose selenocysteine, you are correct. Okay, because again, this question asks for which one is not, okay, the keyword is not a naturally occurring amino acid. So if you go back to the lectures, these are the 20, well, it's written, what is shown here is 21 of amino acid. And according to IUPAC system, selenocysteine is not part of the 20 naturally occurring amino acid. So other than that, everything else is naturally occurring amino acid. So we have alanine, histidine, lysine over there, aspartic, glutamic, serine, threonine, asparagine, glutamic. We have cysteine, glycine, proline, alanine, valine, isoleucine, leucine, methionine, phenylalanine, tyrosine, and tryptophan. Okay, so this image over here, okay, the whole picture will be provided to you during the final exam. Okay, so I will provide this to you, and it's up to you to actually understand and use it if you need to. All right, so meaning that you don't have to memorize all 20 amino acid, as long as you know which one is not the naturally occurring ones, or a variation of that particular question, then you are generally safe. Okay, by the way, if you do need to ask me about anything specific about the quiz or the tutorial, just you can either raise your hand or you can just type it in the chat room. Okay, all right, so number 3, let me see which one is number 3. That one. Okay, number 3, an antibody can bind to more than one target. An antibody can bind to more than one target. True or false? So we will have 1 minute until I will get 19 responses. So far, 11, 12, 13, 14, 16, 17, 18, and 19. Okay. So the answer is false. True. True or false? Okay. 97% of you got it right. So an antibody can bind to more than one target. Okay. So I did not actually specify this sentence during this lecture. But if you recall on when you learn about proteins, enzymes in your biotechnological course, SID3020, okay? So you should remember that enzymes, so to say, is flexible, and in the induced fit model, it's a dynamic process, okay? It's a dynamic process whereby the enzyme moves, okay? Moves to complement its target, okay? So the same thing, what you see here is that an antibody, even though it's not an enzyme, but it is still a protein. An enzyme is also a protein. So protein is a very huge field, okay? So you have that protein, and a subset of this, you'll have enzymes, and another subset is you have an antibody, okay? So because they both of them belong in the same family, in the same group, so they behave rather similarly. Not identical, because enzyme catalyzes a process, antibody does not catalyze any process, not directly anyway, okay? So they have similar function, meaning that they can still move to actually capture its target. So it is specific, but not specific enough that one antibody will bind only to one target. So you can still have an offset target. So this question is actually true. Okay. All right. Question number 4. I'm not sure how this will look like in your phone. Hopefully, you can click the correct area. Where does binding of antibody and antigen occurs? Okay, choose a site whereby the antibody and the antigen occurs. Oh, looks like that. That's nice. Okay, you have 2, 3, 4, 5, 10, 11, 13, 14, 15, 17, 19. Okay. All right. So remember the question? Okay, the question asks, where does binding of antibody and antigen occurs? So the picture, I hope that everybody noticed that this is a picture of an antibody. All right, so it's the same picture. Most of you guys choose over here, some over here, and some over that particular region. Now, if you recall the structure of an antibody, okay, you have the heavy chain, and also you have the light chain. And then what connects between these two chains are, oops, sorry about that, when I exit. Okay, so what connects between those two heavy and light chain is an SS bond, a disulfide bond, which is too small to draw. Okay, so basically you have a disulfide bond over that side, and you have a disulfide bond. I think there's 2 disulfide bond if I'm not mistaken. Okay, so you have 1, 2, 3, and 4. So 2 of each heavy and light pairs. So you have this first pair on the left, which has 2 SS bond, and then you have another 2 on the right hand side that binds the SS bond. At the same time, you have another one, SS bond, that connects between the heavy chains. Okay, so if you remember that, and if you look at here, you should notice that there are 2. If you think about color coding, okay, you can see there's 2. You know that they should be 2 heavy and 2 light. Okay, but if you look at this image, you might not know which one is the heavy, which one is the light chain. So to identify it very, very easily, is by looking at this weird color. Okay, so this color are all a representative of a sulfur. Sulfur, okay, NS. So when you have a disulfide bond, you will see two of this orange color next to one another. So you see two pairs over that side, okay, and over here you don't really see anything. But you do know that based on the structure on this left hand side, you should have two SS very close to one another on the heavy chain, the longest one. So if you look at this image, if I can delete whatever I've just written, okay. So if you look at this image over here, there's no SS in the middle of these two, but there's one over there. Okay, so you know that the heavy chain actually goes like that. So it's like a branching out. So this is the base or the FC region going up, one to left hand side, another one going to the right hand side, and then you have your light chain over here. If you have that, then you should be able to identify that this region are the binding region of the antibody, and therefore this region and that region are the binding sites. Okay, so for those who actually put in the middle one, okay, the middle here or at the base of the FC region, then it's incorrect. Okay, and again, the binding region is not just over that particular site. Okay, it's not just over here. It is actually along this line, everything. Okay, so not just one. All right. Number 5. Let's see, 1, 2, 3, 4, 5. Okay, question number 5. Which of these is the strongest force in a biomolecule? A, static interaction. B, dispersion force. C, covalent force. D, hydrogen bond. And E, none of the above. Six, 7, 8, 9, 10, 11, 12, 15, 16, 17, 18, 19. And do we have 20? 20. All right. I'm not sure why do we have 20. How many people we have? We have 21 over here, excluding me. Should be 19. But anyhow, okay. So for this question, it's kind of like a trick question, whereby I'm asking you what is the strongest force. Some of you, as I expected, will probably choose covalent bond, but some of you will definitely choose hydrogen bond. Okay, so to answer this, oops, I don't have the slide. Oh, I didn't put it in. If you recall the slide whereby all the forces in molecule are actually shown, covalent is not one of them. Okay, so covalent bond is just the yes, it's the strongest bond in a molecule, but when we are talking about biomolecule, it's not just about the covalent bond. The strongest one that we always consider is hydrogen bonding. Okay, so 70% of you got it right. 30% of you almost. Right, number 6. Which one is number 6? Okay, Mr. Rabu conducted an experiment to identify a peptide epitope sequence. The molecule identified is as shown. What is the amino acid sequence of the molecule? Okay, so this is the structure. If you turn it Oops. So this is the structure. If you turn it 90 degrees clockwise, or in this case, it's a rotation towards that particular axis. Okay, from the top to the bottom. If you rotate it like that, then you will get this picture over here. Okay, you will get this one, and then again, if you rotate it from there to that side, then you'll get it over, we will get this picture at the very bottom. So if you look at the pictures carefully, you should be able to identify the sequence. So I'll give you guys 2 minutes to actually solve this. Again, probably it will be easier if this is your real final exam, whereby you will have this list of amino acid. So you can actually look at the sequence in an easier manner. But this is just a test. This is just a tutorial. So I just want to see how you guys will be able to read the sequence. I will just zoom in a little bit in case if you need to see it better. Okay, I give you guys 1 more minute. There's 12 responses now. And scroll a little bit. Ten more seconds. So there's 4 more of you who have not choose an answer. Okay, there's 6%, 78, 18. So 2 more, 19. Last one, 5 more seconds. 4, 3, 2, 1. Okay, so the correct answer is, I'm not sure if it's actually showing. Did it show the correct answer? No, it doesn't. Okay, so the correct answer is, let's go and solve it. Why can't I zoom in anymore? Okay. All right. So if you look at the sequence, okay. So when you're talking about a peptide sequence, you need to know which end is actually the amine, and which end is the carboxyl end. Okay. So the N terminal or the C terminal, C carboxyl, N is amine. So when we write a sequence like this, okay, what it means is that you have your histidine or alanine on the N terminal, and then towards the end on the C terminal, you either have an alanine or glutamic acid or histidine. Okay, and then this one is the COOH, carboxyl acid group. All right, so if you encounter a similar question like this again, the first thing that you need to know is you need to identify where are the N terminal, where is the N terminal, where is the C terminal. Okay, so that's the first thing that you need to do. So if you look at this image, okay, you'll see one carboxyl over there. And then if you scroll up a little bit, you also notice that there's another one carboxyl over there. Okay, so the first image is actually showing you guys that there are two carboxyls. Okay, so now you know either one of these two is the carboxyl. Okay, but because these are all amino acids, so you need to understand the basic structure of an amino acid, whereby you will have, so why can't I delete this? I can't delete that one. Okay, so anyhow, so you need to remember that the basic structure of an amino acid is one having a carboxyl terminal, and then you have your alpha group or the R group over this side, and then you have an amine group on the left hand side. Okay, so when you go back to your structure, doesn't want to detect my hand, sorry. Yeah, how do I do this? Technological error. Okay, so if you look at these two structures over here, so which one represents C terminal better? So of course it should be the one on the right hand side. Why is not this one on the left hand side? It's because if you count the number of carbons, so that's, if that one is the C terminal, that one should be the R group. So this one over here should be the amine, should be the amine group. However, it is not. So because that one is not the amine group, therefore you know that this one is, oops, you know that this one is your C terminal. So that's the first one. Second one, you need to look for the N terminal. And to look for the N terminal is by looking at the blue color at the very end without anything attached to it. Okay, and if you look at the picture at the very bottom that I'm showing you right now, so this is the N terminal. Okay, because there's no, it's not an amide. So if it's an amide, then you see NH, which is blue, and then you'll see a green, and then you will see a green red. Green red signify C double bond O. Okay, which is the carboxyl terminal. So if you see something like this, then you know that, oh, this is the first amino acid, because you don't see anything that represents C double bond O next to the amine group. So because this one is the N terminal, then of course the first amino acid has this as the R group, which is equivalent to a histidine. Okay, so the first amino acid must be histidine. So either A, B, or D are correct. Okay, A, B, or D. So which all of you got it right. Now you move to the second amino acid. So what would be the second amino acid? So you know that this is the structure of histidine, and that histidine is the first amino acid. Then you move through the backbone, and then, oh, that one is the second one. Okay, so that is the first one. This one is the second one. You follow through again the backbone. That one is the third one. Follow through, and then this one is the fourth one. And then finally, you will have this as the fifth one. Okay, if you look at this, even if you can't identify the sequence properly, you should go down and try and figure out. So this one is the, you know that this is the C terminal. So that one is the C terminal. So you can move back, moving backwards. So that one is the first one from the C terminal. Okay, and then you move to here and there, and then moving towards this side, and then you cannot see anything. So you know that this one is second, this one is the third. So in this case, fourth and fifth is a mix, so you don't really know, but you know that from the C terminal, this is the sequence. From N terminal, this might be the sequence. And then you move to look at the last picture. Just delete the whole thing, if I can delete them. Okay, and you move to the last picture. Again, you can clearly see this is the histidine, so which is the first one. Moving up there, and then you will see the second one over here, third one over here, move through the backbone, and then you can't really see the fourth and the fifth, which is fine, because now you can see clearly that from the N terminal, okay, you have one is histidine, okay, second one, which is very clear, this is a glutamic acid, okay, and the third one, it's very clear, it's a leucine. And then because the last image, you can't really see what is the fourth and the fifth, so you go back to the second image. So second image like this, you can see that it is an alanine, okay, even that one is clearly showing you that it's an alanine, so you know the last one is an alanine. And then if you move back to here, you can see another leucine. Okay, so the sequence now is HELLA, and then that one is the C terminal, and H is the N terminal. So if you go through this sequence, then you will see that A is actually the first one. Okay, so the first one is histidine, and then second one glutamic acid, third one is leucine, fourth one is leucine, and then at the C terminal you will get an alanine. So 5% of you got it right. 79% almost got it right. So you got the first three amino acid on the N terminal correct, but the other two is actually wrong. It's the other way around. And then D is also wrong because there's two glutamic acid, and it's written there glutamic acid is at the C terminal, while if you look at the first picture at the very top, okay, it's not a glutamic acid. Okay, all right, so that one is number 6. Now question number 7. Which of these is the main ingredient for a vaccine to work? Which of these is the main ingredient for a vaccine to work? An adjuvant, a carrier, epitope, the danger signal, or none of the above? Okay, which one of these is the main ingredient for vaccine to work? So this one is based on lecture number 4, if I recall correctly. So adjuvant, carrier, epitope, and danger signal. Again, the question asks for main ingredient. Okay, the main ingredient. So 16, 17, 18, one more. One more response? Anyone? Okay. Oops. All right. So I'll just deactivate this to keep the answers. Okay, so which of these is the main ingredient for a vaccine to work? So to answer this question, you need to understand what are the each and individual component is actually is. Okay, you need to remember what is an adjuvant, what is a carrier, what is an epitope, and what is danger signal. And if you don't remember any of that, the fifth option to actually confuse you is none of the above. Okay, so this one, if you go back to one of the lecture number 4, where I showed you what is a vaccine, and the path of an antibody production is antigen processing. So what is an antigen? A minimal component of a vaccine. You need to have antigen, you need to have adjuvant. And then for more advanced analogy, you need to have a signal and delivery or carrier. Now, what is the most important for a vaccine? So for a vaccine to work is one of the pathway is to actually produce an antibody. Okay, and to produce an antibody, what you need is an antigen processing. Okay, antigen processing. Therefore, what you need to do? The answer for this is actually not the danger signal, but it's actually the epitope. Okay, so the epitope is more important than the danger signal. Why do I say that? Okay, now, if you think about the COVID vaccine that we have on the market nowadays. So it's not really the danger signal that produces the antibody. It's not the epitope, because the vaccine itself do not contain any epitope. For argument's sake, we are discussing about the Pfizer vaccine, okay, whereby it's using mRNA technology, okay, encapsulated in a lipid, like that. Okay, so in this particular vaccine, there is no epitope. So to say, directly, there's no epitope in the vaccine itself. There's only mRNA. There's no danger signal. There is no, well, there is a carrier. Okay, the carrier is the lipid surrounding the mRNA. But the technology resolve on the aspect that the mRNA will enter cells, and then it will produce the spike protein, the COVID-19 spike protein. And then that spike protein does not contain any danger signal. Okay, that spike protein is just a spike protein. It does nothing, but because it's a foreign entity, your body recognize it as an antigen, or based on this is the epitope. Okay, so for this question, the correct answer is the epitope. So epitope is a must for any vaccine to work. You might not need a danger signal, okay, as evident from the Pfizer vaccine. There's no directly link to danger signal, but the epitope must be there, because without the epitope, even if you have a danger signal, it will not do anything. Okay, if you have an adjuvant without an epitope, it will not do anything. If you have a carrier without the corresponding epitope that it actually carrying, there will not be anything. So this question, the correct answer is epitope. All right. Okay, number 8. So number 8, what is the critical step for a vaccine to work? Okay, what is the critical step for a vaccine to work? It's very easy. It's very straightforward. You shouldn't write more than 5 sentences. Oh, sorry, 5 words. Okay, it should be less than 5 words. What is the critical step for a vaccine to work? Okay, recall what is the function of a vaccine, how does it work, and what is the critical step of the vaccine, or of a vaccine, for it to work. Okay, without this step, vaccine will not work at all. Anyone? give you guys another 30 seconds. Identity processing. Let's see, we are now on question number 8. Okay, what is the critical step for a vaccine to work? Okay, we have 12 responses, 13. Quick, we have 2 more questions. We only have 5 more minutes. 14. 14? Anyone else? Come on, 15. 4 more. 16. 17. 2 more. 18. And 19. Okay. All right. So these are all the answers. Okay. It is true. So you need to have, well, the critical bit is the end. Then I was hoping that processing becomes larger as well, but there's only antigen. Okay, so it's partly true. So I've already shown you guys this slide. So the part for antibody production is antigen processing. Therefore, the critical step for a vaccine to work is antigen processing. Okay, so antigen is correct. So the other keyword that you need to pair it with is the processing part. All right, question number 9. Briefly describe, this will be long. We'll just skip to question number 10. So otherwise it will be very long. Okay, so number 10. How or what does an opsonizing antibody do? What does an opsonizing antibody do? Does it stop the process of its target? It kills the host of its target, identify its target, then promote phagocytosis, or trigger a self-destruction, or the fifth one, all of the above. Instead of none, so this time around, it's all of the above. You have 4, 50-50. Fifth response, identify target and then promote phagocytosis. You have a little bit more on stops the process of its target. So it's like a race, it keeps on changing. 833, 58, 14 responses. Okay, can I have 5 more? 4 more? Okay, this will be the last question. 3 more? 44? 41? Oh, the risk goes down. 19. Okay. Final answer? All right, so I'll deactivate the question. Now, so again, if you recall, there are 2 types of antibody. If you recall the one of the lecture number 5, okay, in the lecture notes, there is a figure whereby I'm showing you T-dependent and T-independent response, okay, and at the very bottom, there are 2 boxes. One is neutralizing antibody, the other one is opsonizing antibody. So opsonizing antibody kills the host of its target. Okay, what do I mean by its target? Is an antibody. Okay, and this antibody binds to something, so it doesn't directly kills the host, meaning that it doesn't do anything to the host originally. Okay, but if there is a target like that, and then this target has a complementary sequence to the antibody. So the antibody first will bind to that particular receptor or protein or whatever on the surface of the target, and then it will promote the target to actually kill itself. Okay, so this, the most accurate answer is actually kills the host of its target. So the target is this one. So once the antibody binds to it, it will promote self-destruction to the host. Okay, trigger a self-destruction is a close, but it's not the correct answer because it needs to bind to its target first before it actually promotes destruction of the host cell. Okay, so that is all from me. I will post these questions on Spectrum, and I will also post the recorded video or the recorded tutorial on Spectrum. And I thank you guys for participating. And next week we will continue with, what was it? I can't remember. I'll need to have a check whether it will be a synchronous or asynchronous lecture. If I'm not mistaken, it will be asynchronous, but I will let you guys know later. Okay, that is all from me. Thank you, and have a good day, everybody.
Thank you, Doctor.
Thank you, Doctor.
Gitu.